Hi, this is Jack from PhysiologyZone, and in this episode we're going to do the first part of a 5-part series on skeletal muscles. So today we're going to cover the structures of skeletal muscle, and we've split them into microscopic and microscopic features. So starting with the macroscopic features, so on the screen we have a transverse section through a thigh, and to orientate yourself we have your femur, and then we've got some muscles, the quadriceps, the hamstrings and the adductors. So this picture's on screen to introduce two important connective tissues to know about. The first is a superficial fascia. Now this separates the skin from our muscles, and it's composed of areolar connective tissue, which is a type of loose connective tissue, and adipose tissue, which is fat. And then beneath this we have the deep fascia, and that's more irregular connective tissue that lines body walls and wraps around and holds muscles of similar functions together. So if we were to now pull out one of the hamstring muscles and just look at it, you can divide muscles into 3 macroscopic layers. So to orientate you, we have the tendon, deep fascia and muscle belly. And the 3 connective tissue layers to know about is the epimysium, and that surrounds the entire muscle and is the outermost layer. And then we have the perimysium, which surrounds 10 to 100 muscle fibers and separates them into bundles called fascicles. And then we have the endomysium, which separates each muscle fiber from one another. And these 3 layers extend together to form tendons, which attach muscles to bones such as the Achilles tendon, or can form an aponeurosis, which is a broad, flat tendon sheet. So now let's move on to the microscopic features of skeletal muscle. So we're going to start from the muscle fiber, which is where we left off, and break that down into its constituent components. So here we have our muscle fiber, which is surrounded by the deepest part of connective tissue, the endomysium. Now the muscle fiber is tiny. It ranges from 10 to 100 micrometers in diameter, and it's about 10 centimeters long. So it's like tiny piece of string. And muscle fibers are often referred to as muscle cells, and it's important to know that this is an interchangeable term. And they're the most important aspect of muscles, because within these are the units that allow contraction and relaxation of our muscles. And interestingly, the number of muscle fibers that you have is predetermined before birth. And that's important because when muscles grow, they do so by hypertrophy, meaning the size of the actual muscle fiber, versus hyperplasia, which is the number of muscle fibers. However, as we know, when muscles do repair themselves because they're damaged, such as exercise or otherwise, then we do lose some muscle fibers. So there's obviously something that's there to help regenerate these, and that's due to the presence of satellite cells. And these are effectively stem cells for muscles, so that they allow our satellite cells allow muscle growth through our life and repair our muscles when they're damaged. So next, within the muscle fibers, we have many little strands, like little pieces of string, that are called myofibrils. So just pull one of those out. And underneath the endomysium, you have a cell membrane of the muscle cell or muscle fiber, which wraps around all of the muscle fibers, called the sarcolemma. And right underneath the sarcolemma is the nuclei, which makes the proteins for contraction, and we also have all of our myofibrils, which are the units that cause the contraction. And then from the sarcolemma we have a series of hollow pipes which come down to the very center of the muscle fiber, and they wrap around each myofibril, and these are called transverse or T-tubules. And because they're open at the top, then they have interstitial fluid in them, which is important to allow the electrical current, which is the action potential. So obviously the sarcolemma is a cell membrane, so outside of that is the extracellular space, so that has interstitial fluid in it, and that's the salty fluid, and that can move down those pipes, and then when the electrical current comes, it can pass over the sarcolemma and get down through the T-tubules. And then next to your T-tubules, you have a series of sacs that are around the actual myofibril that's called the sarcoplasmic reticulum, and at the end of these are actually dilated, and that area is known as the terminal cisterna. And these terminal cisterna are in contact with the T-tubules, and where two terminal cisterna meet with one T-tubule, this is known as the triad zone, and it's important to allow the action potential to get from that T-tubule all the way across all of the myofibrils. And then underneath our cell membrane, our sarcolemma, and surrounding all the myofibrils is the cell fluid of the muscle cell, which is called the sarcoplasm. And just note that in any other cell, this is called cytoplasm. And within this, there's quite a few things, but there's three key things that are there that allows muscles to contract. There's glycogen, which is just a glucose polymer, and that's used to create our energy in the form of ATP. There's also myoglobin, which is a protein which is only in muscle, which binds oxygen in the surrounding fluid, and then it releases it as is needed to the next structure, which are the mitochondria, and these lie in rows in the muscle fibers, and they generate our ATP for muscular contraction. So in the last episode we talked about this, which is the microscopic features of skeletal muscle, and if you remember, these were that the fibers lie parallel to one another, they've got multiple nuclei on their outside, which as we remember now is just underneath the sarcolemma, and the fibers themselves appear striated. Now the reason for these striations is due to the presence of the sarcomere, which is the basic functional contractile unit of the myofibril. Now, the sarcomere is a series of structured repeating units which makes up the myofibril. And now if we just isolate a single sarcomere from the myofibril, we can then go through its structure. So it's made up of two filaments, thick filaments and thin filaments, and they're layered in the sarcomere in a 2-to-1 ratio. So you've got two thin filaments for every thick filament. And now we can go through some of the boundaries within the sarcomere. So firstly, there are Z discs, which are also known as Z lines and Z bodies. This is just dense material that separates one sarcomere from another. It's easy to remember because they look like Zs. And then we have the A band, which is the thicker middle bit of the sarcomere, and it's darker. And then we have the I band, which is the lighter, thinner outer portion of the sarcomere. And you can note that the I band doesn't finish at the end of the sarcomere, it continues all the way on until you get another A band. And the A band is also called the dark band, the I band is called the light band, and this is what gives the stripy appearance when you look under a microscope at skeletal muscle. The reason why they're called that is just because of the way that they look under the polarized light. So A stands for anisotropic to polarized light, and I stands for isotropic to polarized light. So next we have the M line, which is the middle line of the sarcomere, and it contains supporting proteins that hold those thick filaments together in the middle. And then obviously we have to have another zone, a H zone, and the H stands for heller, which is German for brighter, and it's within the A band, and it's just the portion of that thick filament which is not overlapped by the thin filament. So if you will, it's sort of the slightly lighter bit of the A band. So now that we've looked at the area of the sarcomere, let's consider what it's composed of. It can be broadly split into three groups of proteins. So firstly, we have the contractile proteins. We have actin and myosin. So myosin is part of the thick filament, and it's the predominant part, and it's shaped like two golf clubs that are twisted around one another, and it's made up of heads and tails, and the tails lie parallel to one another, and they point towards the M line in the center of the sarcomere. And then we have actin, which is the main part of the thin filament. That's anchored to the Z disc on the outside, and the actin molecules are twisted into a helix formation. And on each actin molecule we have a myosin binding site, which is for the myosin head during contraction. Then we have some regulatory proteins, and there's two important ones to know about, and they're both on actin, and these are tropomyosin and troponin. Now firstly, troponin is actually made of three proteins, troponin C, troponin I and troponin T, and that holds the tropomyosin in place. And the tropomyosin's role is to regulate when a muscle contracts. So in a relaxed state, when you don't want muscle contraction, it does this by blocking the myosin binding heads on the actin, and then when contraction does happen, it moves out the way to allow that, and we'll go through that in the next episodes. Finally, we have some structural proteins, and their role is purely stability, so they maintain the shape and the elasticity of the myofibrils and thus the overall shape of the muscle. And I just want to mention a couple very quickly. So on the thick filament we have titin, which is from titan, meaning gigantic, and that's the third most abundant muscle protein behind actin and myosin. And its role is just to stabilize the position of the thick filament by anchoring it to the M line and Z disc. And it's also got some roles in elasticity because it's very, very flexible and it can stretch about 4 times its original length. Myomesin's also on the thick filament, and that forms the M line, which connects adjacent thick filaments to one another. And then on the thin filament we have nebulin, which is just a non-elastic protein that wraps around the thin filament, and it keeps its shape and it aids its anchorage to the Z disc. And then finally, I just wanted to mention dystrophin, which is a muscle protein which, with other regulatory proteins, this is involved in reinforcing the sarcolemma, and its only real relevance is to know that it has a very intimate link with a disease called Duchenne muscular dystrophy. Now, this is an autosomal dominant X-linked form of muscular dystrophy, so it only affects males, and it's where the gene encoding for this protein, dystrophin, is mutated. And therefore, if the stability of this protein is compromised, then the sarcolemma's stability is lost. And through a series of cascading events, it leads to a much easier tearing of muscles during contraction, and it ultimately leads to muscle fiber death and replacement of this with adipose and connective tissue. So that's everything that I wanted to go through on this episode. And so let's just recap what we've gone through. We've looked at superficial and deep fascia. And then we've looked at those 3 connective tissue layers, the epimysium around the muscle, the perimysium wrapping it into fascicles, and the endomysium around the muscle fibers. And then we moved on to the microscopic features. So we saw the cell membrane, the sarcolemma, which is around each muscle fiber. And then from there we have the T-tubules, and around each myofibril we have the sarcoplasmic reticulum, and then they dilate into the terminal cisterna. And where those T-tubules and terminal cisterna meet, we have the triad zone. And then we looked more at what the myofibril consists of, which is these repeating units called sarcomeres, and that's made of myosin and actin. And on actin, you have the tropomyosin and troponin, which are those regulatory proteins that are intimately involved in contraction. So thank you very much for watching, and we'll see you in the next one.